So I'm a PhD student at the Innovative Immunotherapy Unit from Raffaele Scientific Institute in Milan. And the main focus of my work is to develop a preclinical mouse model which is able to recapitulate the pathophysiology of ICANS, CAR-related toxicity. We know indeed that CAR T-cell therapy carries the risk of significant toxicities such as CRS and ICANS. And ICANS has been reported with different CAR specificities, costimulatory domains, hematological and solid tumors. Unfortunately, the pathophysiology of ICANS is still poorly understood. And this because the preclinical mouse models are limited and the main, I can say, information come from autopsies performed on brains of patients who suffered from fatal neurotoxicity, without also a direct comparison with mild or reversible cases. So the aim of my work is to exploit our humanized mouse model, which has been developed and recently published, and which is able to recapitulate the CRS CAR-associated toxicity. In this model, we were able to identify some multifocal brain hemorrhages, which are reminiscent of clinical neurotoxic manifestations observed in patients who experienced a high-grade ICANS. Moreover, we were able to assess the distribution and the incidence of these multifocal brain hemorrhages, and we found that the brain regions which are mainly involved in these neurotoxic manifestations are those deeper, involving regions such as the pons, the trunk, and in particular the cerebellum. And especially in the cerebellum, we were able to assess that there is a greater involvement of the molecular layer, which we know is located by neurons, which then are involved in shaping the spike activity of Purkinje cells, which then participate in processes such as motor control or learning features. So we try in some way to assess the clinical relevance of the phenomena observed in the mouse brains. And we found changes in locomotor activities of our mice, performing, for example, beam test walking and assessing their capacity to correctly move and to learn the tests that they will have to perform. At the same time, we were able to identify hypointense lesions through magnetic resonance imaging, which once again are in some way associated with the hypointense lesions observed in patients experiencing high-grade ICANS. And we had also the opportunity to see an extravasation event, suggesting an increase of blood-brain barrier permeability. We know indeed that has reported in literature, one of the main features associated with the CAR-related neurotoxicity is indeed changes in vascular permeability. And by performing an in vivo permeability assay through the Evans blue azo dye, we were able to find an increase in the nanogram of Evans blue injected in the bloodstream per milligram of brain tissue of mice treated with CD19 CAR T cells. The main advantage of our humanized mouse model is that it is able to recapitulate CAR-related toxicities after treatment with human CAR T cells. Indeed, these toxicities are emerged following the interaction of human CAR T cells, human immune cells, and tumor cells, suggesting that this is a great model to recapitulate features that we observe in patients.